Okay, so before I really get into this marker gene profiling lab, I just wanted to kind of give a couple of points, I guess, about earlier. I'll also note that on the projector, TV, whatever, there is a link to the Zoom in the Slack channel. So if anyone wants to join and look at the slides on your own laptop, you can. Also, the slides are on the website, so you can look at them there as well if it's difficult to look at them on the screen. And I wanted to say that if you didn't make it through everything this morning, that's fine. None of the workshops are really dependent on you getting through the previous one. Obviously, there's always a bit of a balance between making sure that we have enough content for anyone that needs it, that gets through everything really quickly, but also making sure that it's not too overwhelming for those of you where it is your first time doing anything like this. So don't feel panicked if you didn't get through it. The instances, if you're in only the beginner workshop, the instances I think will be kind of finished tomorrow afternoon after the workshop. If you're in advanced as well, it will be the same instance that you have all week. So it is also available on Wednesday. There are also ways that you can get hold of an instance after if you want to be able to use one. And we can post some details of that in the Slack channel, but basically there's some instructions for you to be able to use these for yourself. Anyone have any questions on that before I get going? No? Okay. So with that, in this lecture, we're hoping that by the end, you will understand amplicon sequencing, be able to differentiate different amplicon targets, be able to contrast variable regions within particularly the 16S rRNA gene, but also some of the other genes. Hopefully you'll understand when we might use different amplicons to characterize a microbial community. You'll be able to outline the major bioinformatics steps in amplicon data analysis and understand the basic outputs from processing amplicon data analysis. And so I wanted to I wasn't sure whether I should call this a disclaimer or a preface, but I just wanted to say that for most of this, you shouldn't always just take our word for it. Similar to what Morgan has said, you should always do your own research and decide whether you think the methods that we've used are the best for whatever your purpose might be. There are often lots of different ways to do things in bioinformatics. Sometimes you might feel that there is a best way. Sometimes there is no single best way. You just need to research the options, make a decision, and make sure that you can justify that decision to reviewers, to your defense panel, whatever it might be. And sometimes there's just only one tool that you're going to be able to make work. Or maybe there'll be no tools that you can make work. Sometimes bioinformatics is really a pain. So yeah, there are different methods and they may or may not be the best for the job. And often these different methods will make different assumptions about your data that may or may not be correct. But also then it's kind of important to have conversations within yourselves with other people. It's always useful talking to people and hearing about new approaches. There might be a way that you've done something for years and years, and you may have just completely missed a new method that's actually come in and is doing a way better job. So yeah, bioinformatics is definitely kind of constantly evolving, and it's important to stay in tune with that. While obviously it's also nice to kind of keep things standardized between maybe different projects or whatever that might be. Yeah, so moving on to methods for studying the microbiome. We kind of divide these into two major topics. We have amplicon-based and we have metagenome or metatranscriptome-based. So amplicon-based, we're typically looking at 16S, 18S, or ITS, but I will talk about a couple of other genes that are typically used as well. And so an amplicon is a piece of DNA or RNA that is the result of amplification via PCR. And we're typically wanting to sequence a universal gene or barcode, something that we're going to be finding in a lot of different organisms, so that we're able to get information about our community typically. And so we can use these to identify the taxa in a sample, or it might be that we want to look at the diversity within a function. And usually we're needing to be able to design relatively universal primers for that, while hopefully having some variation within those sequences so that we're able to look at those different taxa that are there. And this means that it's kind of restricted to the organisms that actually contain the gene that we're targeting. But host contamination isn't usually an issue here. When we look at metagenomics or metatranscriptomics, we're sequencing everything that's in the sample. So we're not using PCR to amplify something first, but it means that we can get at both kind of who is there as well as what are they doing. And this means that host contamination or high diversity can mean that we need really high read depths. And it also makes comparison with databases much more complicated as we have many more unknowns. But during the beginner workshop, we're going to be focusing on the Amplicon-based analyses. These are at least kind of slightly more standardized in terms of the major steps that we're typically using for bioinformatics. There are a lot more options for metagenomics, and anyone staying will kind of see those later on, I guess. So this is kind of the same comparison that Morgan showed earlier, just kind of demonstrating that there are massive differences in the pricing for these different options. And that's obviously reflecting the amount of data that you're going to get out. But this doesn't mean that metagenomics is always going to be best. Sometimes Amplicon-based is actually going to give you much better information, particularly when you have very high host contamination. If anyone's looking at, something like human oral samples or heard about cancer samples, like tumor samples, most of the DNA within that sample is going to be human reads. And so if you're doing metagenomics, you're going to have like 99% human reads within that, which means that you're having to sequence. If all you're interested in is the microbial community, you're having to sequence a very high depth to be able to get not many microbial reads. And in a case like that, Amplicon may well be really your only option. And so this kind of brings us into ribosomal RNAs. I imagine most of you know about ribosomal RNAs, but just as a recap for anyone that doesn't, these are non-coding RNA, which are the primary component of ribosomes within cells, and they're essential to all cells and therefore present in all living organisms. They play roles in the processing and translation of proteins, and they're kind of organized into a small and a large subunit, usually called the SSU, small subunit, and LSU, large subunit, and one of each forms the ribosome. In prokaryotic ribosomes, we have the 23S and 5S rRNA in the large subunit, and that's about 3,000 base pairs in total. And we have the 16S rRNA in the small subunit, which is about 1,500 base pairs in total. In eukaryotic ribosomes, we have the 5S, 5.8S, and 28S in the large subunit, and that's about 5,500 base pairs in total. And the 18S in the small subunit, which is kind of 1,800 to 2,000 base pairs. And it's a bit more variable than the 16S rRNA gene is in length. These tend to be quite conserved over time due to their kind of crucial role in ribosome function, and they're therefore rarely horizontally transferred, which is what makes them really useful for phylogenetic analyses where we want to be able to get at taxonomy, because them being relatively conserved over time means that their divergence is often somewhat in line with kind of general species divergence. And so yeah, they are what you will most often see in Amplicon analyses of microbiomes. And we can also use them to build phylogenetic trees. This works better or worse depending on which one you're looking at. And often you'll see phylogenetic trees that are also made up of multiple genes kind of added on together to each other. In terms of the marker genes that we use for bacteria and also archaea, I probably should have put that, we're often looking at the 16S rRNA gene. That's by far the most commonly used in microbiome analyses, but there are other genes that people will use as well, and some of this depends on what your purpose is. Some of them will work better or worse in different systems. And also, for example, if you were interested in something like nitrogenase or ammonia monooxygenase activity, you may be looking at sequencing one of these genes so that you can look at the taxa that are contributing to that function within your samples. So while typically Amplicon analysis is not giving you a whole lot of functional information, you can still get at the taxa that are contributing to a function like that. When we're looking at eukaryotes, the two major genes that are sequenced are the 18S RNA gene or the ITS2. Probably, I'm never quite sure if RNA is the right term to put in here, because it's kind of the region in between some of it, but it is kind of part of the ribosome. And yeah, for viruses, there's really no universal marker gene. So the 16S RNA gene, which I'm going to focus on mainly in this because that's what we're doing the most of, but you will see in the lab that we have the option to look at the 18S or ITS2 genes as well. The 16S RNA gene contains 9 hypervariable regions. So if we look at this graph that I've taken from this study, you can see that this is about 1,500 base pairs long, usually, and the measurement is typically based on the E. coli 16S RNA gene, but it doesn't vary a huge amount between different taxa. So what we're looking at on this graph is these kind of highly conserved regions, these kind of valleys in the graph, they're interspersed with these highly variable regions, all of these peaks. And so you can see that we have variable region 1 to 9 along here. And what we really want to take from this is that because we've got these highly conserved regions in between, this allows us to design primers that are relatively universal and will work for quite a lot of different taxa, but we're going to be looking at sequencing one of these variable regions so that you can get a lot of the diversity within your communities. But because they have different amounts of variation, they will give you slightly different community composition results. And you can see this is taken from the IMR website. This is just an indication of some of the different variable regions that we might sequence, as well as the different taxa that you're able to pick up with each of these. So one of the most common Amplicon regions is the V4, V5 region. And you can see that this gives you coverage of approximately 85 to 95% of bacteria, similar percentages of archaea, 81 to 93%. But you also need to bear in mind that you are also sequencing eukaryotic and mitochondrial or chlorophyll DNA within that. And so, you have these other ones that are kind of bacteria specific or archaea specific, where you're actually able to sequence, although you're not getting quite as good coverage of the bacteria, you're able to sequence more or less only the bacteria, which, depending on what your question is, maybe that's of use to you. And you can see these for a bunch of different Amplicon targets here. And so this is just an indication of the differences between those regions. So this is taken from the same study again. And in this study, they took a bunch of 16S sequences where they knew which taxa they came from. And they kind of chopped those sequences down to just those variable regions that they were looking at. And they looked at how well they could then classify the taxa back to what they came from. And so if you're looking at the full 16S RNA gene, you can see this is almost all white. And so that means almost nothing was unclassified. So they could classify almost all of it. So you get quite good taxonomic resolution with the entire 16S RNA gene. But if you're looking at these other regions, you can see that as we're getting a lot more red, so like the V4 region, you're actually leaving quite a lot of the different taxa unclassified in this case. This is kind of important to take into account when you think of something like the Earth Microbiome Project that recommends that you use the V4 region primers, but actually you're not going to be able to classify a lot of the taxa in your sample, particularly down to species level. And then this is also just another comparison, just showing how this looks after you've sequenced a community. So this is looking at a mock community here. I think it has about 20 different taxa in there. And these are 3 different replicates, in each case using the V4, V5 region or the V6, V8 region. And you can just see how the composition of these different groups changes. So if we look at this red one, the Neisseria, you can see that you start off with probably 5% of the community. If you're looking at the V4, V5 region, this is probably more like 15% that it ends up as. And the V6, V8, it's kind of more again. So obviously, everything that you're looking at when you're looking at Amplicon sequencing is kind of imperfect, but understanding how these choices that you make will impact what your results are at the end, I think is important. And so, yeah, if you look at some of these really big projects that have kind of aimed to standardize the target that you might be looking at, if we look at the Human Microbiome Project, they sequenced a lot of different samples. And they sequenced them with different regions, as well as many of them with metagenomics as well. And so they did V3, V5 for all samples, V1, V3 for about 3,000 of them, and V6, V9 for about 100 of them. And then looking at the Earth Microbiome Project, as I mentioned, they're using the V4 region. And so, as I mentioned, these might not necessarily be the best targets, but if you are wanting to be able to compare your data with some of these previous projects that are out there, then you may need to kind of opt for something that may be less optimal for the sake of comparison with something like this. Okay, so then briefly, just to go into the 18S rRNA gene and why you might use it. It's, as I mentioned, a component of the SSU in eukaryotes. It's a homologue of the 12S in mitochondria or 16S in bacteria and archaea. On average, it is 1,800 to 2,000 base pairs. You will find, though, that where in 16S sequencing, you can typically trim everything to a similar length, you have a lot more variation in your 18S RNA gene. So when you're looking to trim to kind of 300 to 500 base pairs or something, that's usually what we can sequence effectively with short read sequencing. In 16S, everything will end up more or less the same length, but that's not the case for something like 18S or ITS. So it's kind of just worth keeping in mind so that you don't panic that something's gone really wrong with your data processing when it's kind of just an artifact of the different gene that you're using. And the 18S rRNA gene also has 9 hypervariable regions. And so similarly to the 16S, these might give you better or worse taxonomic resolution, might be better or worse for different applications. The ITS gene is the internal transcribed spacer. And so it's a spacer unit between the SSU and the LSU in rRNA. Bacteria and archaea both have a single ITS unit between the 16S and 23S, whereas eukaryotes have 2. So the ITS, or ITS1, it's often called, is between the 18S and 5.8S units, and ITS2 is between 5.8S and 28S. And so sometimes now, as we're moving more towards longer read sequencing, particularly, the whole thing might be sequenced. And so in that case, we're looking at the ITS1 plus the 5.8S plus ITS2, and that's about 550 base pairs. But the ITS2 in shorter read sequencing is what has been used the most often, typically. And that's because it has a more conserved secondary structure than the ITS1. But again, this is more variable in length than the 16S. It's much shorter, but you're seeing very large amounts of variation in the length between different taxa. And so, yeah, just to give a quick indication of the full-length sequencing, we're not doing it in this workshop, but as Morgan kind of mentioned earlier, it is now much cheaper than it used to be, and it's also much more accurate than it used to be. So people are moving towards this more and more. And there's not typically as much variation in the different primers that people will use to sequence them. Okay, so I'm not going to go into Illumina sequencing in detail, but just kind of important things to remember when we're looking at going into our bioinformatic processing steps is that we are usually going to have a first round of PCR where we're doing a taxa-specific PCR. So that's where we have the primers that we've just been looking at here that are going to hopefully work on a large variety of the taxa that you're interested in. And then we have a second round of PCR where we're attaching the indexes that Illumina is going to be looking for to sequence. And then when we have this kind of final PCR product that is your kind of the area of the region of interest in this darker blue color in the middle, your taxon-specific PCR with the gray, and then the index primers in the blue and the yellow, all put together, we tend to call this a library. And the library will then be the pool of all of your samples together. Oh, and I should mention the indexes are specific to each sample. So you're going to be able to map each of your reads back to the sample that it came from based on these kind of short sequences that are added onto that DNA product. So when we come to look at different options for marker gene analysis, you will find now that QIIME2 is by far the most used for this. And this is partly because it's got a really large team of people behind it that are developing and maintaining it constantly. Mother was more used historically, and it definitely can still be used now. But it's kind of more or less one guy that's developing and running it, which I think probably you're going to, like, QIIME is always having new methods added into it, different pipelines. It's kind of continually under development, and there's probably tens of people involved in the upkeep and maintenance and development of it. And so I think it's just going to naturally have a lot more options and be kind of more utility for different things. But that doesn't mean that it's any better necessarily than something else like Mother. But it tends to be kind of the most user-friendly. It's what most of the people use for analysis now. And so it does also mean that there's a lot more help available for it. There are other options. So the Data2 pipeline, this can be run directly in R. The other, QIIME and Mother, both run from kind of command line. But Data2 can be run in R. There are also other options like Microbiome Analyst. This is kind of a web-based system where, you know, maybe it would be great for someone to use if they're going to do one project. They maybe don't need so many options for customization as you get in a command line option, and it has quite a few kind of built-in statistical profiling things. I haven't really tested it that much myself to know how long you might have to wait to be able to run something, because obviously it's someone else's server on the back end that's running it. And so if you're trying to run a large number of samples, you potentially might be waiting a long time for that to happen. And there are other options as well. There's you know, new papers coming out all the time with options. I haven't looked into Lotus 2 a whole lot, but you know, there are always more options. As I mentioned at the beginning, you should research which things are out there and might be best for your kind of particular purpose. But we today are going to be focusing on QIIME 2. It's what I use all the time, and most people that are regularly doing bioinformatics will be using the most. So QIIME, this actually may be an old image now. They've just recently overhauled all of their website, actually. But basically they have a lot of different material, tutorials. They have a forum. They have tons of different options for getting help and for finding what you might be wanting to do with your data. And one of the nice things about QIIME is that it does kind of track which steps you've already run. And so you can look at that in their web browser. You can see what you've actually done to get to the point where you've got a file that is a QIIME file. And with that comes that they have these kind of two major file formats. They have a QZA file, and this is a file that's made up of your data, essentially. So when you start with your raw reads, you will read this into QIIME. It will become a QZA file. And that is what QIIME will perform each of its analysis steps on. And QIIME then has a second file format that it uses, a QZV file, and this is a visualization file. And so this is how you can really easily tell when you're looking at the files that you've made throughout your analysis. The .qzv is something that you'll be able to visualize. You can add it to their website, this QIIME 2 view page. And that's where you'll be able to look at information. I mean, similar to how you would have looked at the multi-QC or the fast QC files earlier. It's kind of a more interactive way of looking at it than just on the command line where you're looking at text. And so QIIME also allows people to develop more plugins, they call them. And so this is nice for, you know, people that come up with new methods, new algorithms for different processing steps. It means that they can add these into QIIME. They're fairly rigorous usually with making sure that they will be able to be easily installed by other people. And there's definitely very fast support available through their forum, so it is quite nice to use. And now with that, we also, well, this was even before I was working with Morgan, a long time ago now, there's a Microbiome Helper paper that came out, which kind of gave an initial kind of workflow for processing your DNA sequencing data. And this actually also included the wet lab steps for generating it as well. This was published in 2017, and as you can imagine, many things have evolved since 2017. And we have a GitHub that's associated with this that has a bunch of Wikipedia pages, and you can see the kind of current steps that we use in our lab for a lot of different workflows. I am in the process of trying to update this and eventually publish the Microbiome Helper 2 paper that will have things that are much more up to date, but it is kind of a living document. We update it as it needs updating, as there are different QIIME 2 versions, things like that, other tools, anything that we're looking at kind of, whenever we're standardizing pipelines within the lab, we tend to add them onto there. And it's kind of a quick reference for each of us, as well as anyone else that wants to use it, because it's freely available. And there is an associated Google group where people can kind of post and ask questions about analysis and things like that. Okay, so what we will be doing this afternoon, and a kind of typical bioinformatics workflow for Amplicon data is we're going to be taking our raw sequencing data, FASTQ files. We will be pre-processing them. So some of this is already done by the sequencer in the case of Illumina data. And so that's kind of the way that some of it is today. So the demultiplexing, so that's taking it from kind of just all of the data that has come off the machine, all of the reads from everything in that run, and kind of taking it back to the reads that belong to each of your samples. That's already done. We don't need to worry about that. But what we will be doing is removing the primers, joining the reads together, the forward and the reverse reads, and quality filtering them. After we've done those steps, the next step is usually denoising. And I'm going to explain a bit more about OTUs versus ASVs in some of the next few slides. So for anyone that has that question, just hold off a second. But yeah, so after the denoising step, we then hopefully have some representative sequences. And at that point, with those sequences, we can do a couple of different things. We often are looking to do taxonomic assignments. And from that, we will usually then build an ASV table. Once we have that, we can do kind of downstream analysis and statistics and visualization. And the second thing that we often do with those sequences is we align them, and we're often wanting to build something like a phylogenetic tree with them. We're not going to be doing that today, but you will be doing some of that tomorrow morning. And so with this, we also then have a metadata file, usually, as Morgan talked about earlier. That's not really going to come into it today, but again, it will be coming into it tomorrow morning, where you'll be kind of looking to combine this metadata table into some of this downstream analysis and visualization. So sample demultiplexing. So this plate on the right-hand side, this is kind of a typical Illumina setup where you have 8 different primers for indexes for your different rows, and you have 12 different primers for the different columns within this. And these kind of 8 by 12 combinations mean that you have a unique index for each of your samples. So you don't really need to worry about this, because this is what the Illumina sequencer is doing for you. But it is kind of useful to understand that that's where each of the samples comes from, and that's how you can kind of just combine the DNA from all of your samples together when you're sending something off for sequencing. And kind of seems crazy that you're just sending off this like 20 microliters of liquid to be sequenced for all of your hundreds of samples potentially. But yeah, each of these will have a unique barcode to link back to it. People will sometimes combine different amplicons into the same sample. So if you know that you're not going to have much overlap between amplicons, so you could say have 16S and 18S and the same index for each of them, because within your 16S processing, you'll probably be filtering out the eukaryotes. And in your 18S sequencing, you're going to be filtering out any prokaryotes that you've found in there. So people sometimes do some different things like that. It all ends up working fine in the end, typically. And then Morgan already went through quality filtering, and you've done some of that this morning. So I won't labor on this point. Only that you will notice in QIIME, so it's a slightly different visualization that you'll see for the quality of each of your sequences. But as we discussed earlier, the reverse reads do always have a lower quality than the forward reads. just kind of affected the machines, but it's just worth kind of bearing in mind when you're looking at them again, that it's not a panic that they have lower quality than the forward reads. So then we have rejoining, and this is where we're joining together the forward and reverse reads. Again, Morgan kind of touched on this, but when you're thinking about a forward read and a reverse read that are each about 300 base pairs long, we're typically looking for about a 550 base pair PCR product. I mean, if that's kind of V4, V5 region anyway. And you will also have a certain length kind of for the primer sequences that you have in there as well. And what you want to do when you're designing these primers is make sure that you have enough overlap in the middle that you will be able to join those reads together, if that's what you want to do. I'm really like, this is not really based on science. This is based on my feeling, maybe like 25 base pairs at least. That seems reasonable to me. I think there's a lot of steps in this that you'll find we get a bit like hand wavy. And you just, after you've looked at a lot of data, you kind of get a feeling for like what's normal and what's not normal when you're looking at things. And I know it often feels a bit frustrating when you're learning things that people are like, oh, how did you know that's the right thing to do? And you're like, it just looks good. And I guess something like the V4 region would have pretty good overlap, but I mean, it just depends, because at that point you're sacrificing your kind of taxonomic resolution. And I guess it depends what's most important for you. Probably some more overlap made a lot more sense when sequences were not as accurate, because they've been getting more and more accurate. If you're going to have fewer and fewer, you know, because if you're overlapping, it helps with any errors that you have in the sequencing. If your errors are like, 0.01%, that's impacting you a lot less than even, 1% or something. So yeah, I do think that as the sequencing technology had got better, it probably matters less. And also as the algorithms for bioinformatics have got better as well, it probably matters less as well. But yeah, it's probably one of those things that with a bit of a trade-off, it depends what you're most interested in and what matters most for your project. Yeah. Yeah.
When you have complete overlap, does that mean the sequencing reads are getting shorter?
Yeah, because I mean, essentially, if you only have 300 base pairs in each direction, that limits you to kind of a total of like, say you're looking for almost 290 base pairs overlap there, that's your maximum length. And if you're trying to assign taxonomy to that, then you've only got 290 base pairs of kind of information for doing that with. Whereas if you have a longer one, you're more likely to see kind of variation between different taxa that allows you to assign it to a lower level. Right. Yeah. 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 Okay. Yeah, and so then usually we're using vSearch within QIIME 2 to join these together. That's the name of a program that joins reads. There are other options. Pair is one of the ones that's often used more for metagenomics. But yeah, I'm sure there are many other options as well. I don't know that I've ever really looked and compared at them. Some of them seem to do better jobs at dealing with errors between some of them. Maybe that's a good thing, maybe that's a bad thing. It depends. But yeah, there are many options. Okay, so denoising is probably the kind of biggest step here. So traditionally, I would say up till about probably I want to say data was probably 2016. So up until about that point, people were doing OTU clustering. And so an OTU is an operational taxonomic unit. And it's often kind of been thought that this was approximately equivalent to a species level. Usually these are being clustered at about 97% similarity. And it's important to remember that's 97% similarity within the gene that you're looking at, not necessarily the genome as a whole. And there are different options for this. You have de novo clustering, where this is just looking at the reads within your samples and trying to cluster them together at 97% similarity. You can have closed reference clustering, where you might be giving a database of sequences and you're requiring each of your sequences to be kind of 97%, say, if that's the threshold you choose, similar to those sequences. And you also can have open reference clustering, where you're kind of preferring the sequences in your sample to be clustering to those sequences in a database already, but you're also allowing them to just cluster with other sequences within your samples. And typically, if you had samples from a very well-characterized environment, like the human gut, the closed reference clustering might well work quite well. You're probably going to be able to cluster most of your samples that way. But if you're looking at like deep sea hydrothermal vents, you're probably not going to be able to cluster many of your sequences to the sequences that already exist in the database. So the more kind of unknown your environment, the more likely you are to need kind of something more like the open reference clustering. And of course, clustering at something like 97% means that you're losing information, because that kind of 3% diversity between the sequences, you're just kind of ignoring it exists. So you lose information, but also at the same time, trying to collapse at 100% means that one single base pair error in your sequence would mean that you've kind of assumed that you have this new taxon that actually doesn't exist in practice. So then people started coming up with the ASV methods, so Amplicon sequence variant. And the idea here is that they kind of create a model of the errors that they think exist within your data. I can't go into detail of how they actually do that because it's kind of complicated. But essentially, we kind of trust that they are doing a decent job of correcting the errors that are introduced by sequencing, because these errors tend to be relatively kind of uniform within different sequences. And there are different methods for doing this. Data2 was one of the first that came out, and the names I've given here are just kind of the kind of key people that went into developing these and the different groups that kind of advocate for their use. So some of the biggest ones were Data2, dblur, and unoise. I would say now Data2 and dblur are the two that are most commonly used. I think they may be the only ones available in QIIME2, not 100% sure on that, but they are definitely, yeah.
Those ASV models, are they, you mentioned they're platform specific, are they available for Illumina, PacBio, and were they just presumably?
Yes and no. Yeah, so definitely for Illumina. PacBio, I believe you need to use Data2 for. It has an option. I could have that the other way around. It could be that you need to use dblur. But yeah, Illumina, almost everything will work on, I guess, because it's just the most common that everyone's developed for. Depending on what else you're looking at, yeah, you might be limited as to what you can use. And I guess that goes back to my idea of, you might have decided one of these is best, but if it's not available for the platform you've used, then you're stuck. But I don't have it. I may have a slide in a couple of slides time, just with a comparison. Essentially, we don't find it makes that much difference usually which of these you choose. I'm sure, every time a paper is published introducing one of these, they show how it's better than everything else that's ever existed before. And maybe it is in this kind of one special application, but often in practice, we don't find that there are actually that many differences. And, you might end up with slightly different numbers of ASVs at the end. Maybe if you're looking at the rare things, you will have some more differences there. But typically in the dominant taxa within the community, we don't really see differences regardless of which one you use. So yeah, this is kind of just a bit of a visual representation of the difference between OTUs and ASVs. So if we imagine that we have each of these in our kind of sample, each of these blobs is representing a different real sequence, and the size of them is kind of the abundance. you might imagine that when we're sequencing and we're introducing these errors, we're ending up with these kind of all of these different options, even though this one, kind of looking at this blue one, this one might be what actually the sequence was supposed to be. And so if we're looking at OTUs and we just cluster a certain percent similarity, we might be losing some of the information between, you know, these two things that actually were different from one another, although they may fall within our kind of 97 or 99% threshold, whatever it is that we set. But hopefully with ASVs, you're hoping that you're getting a bit closer to what this real sequence was, but it doesn't mean that it's perfect. You're probably still going to end up with some extra things, but at least less so. I mean, in my own kind of practice, when I've seen these, I was often finding that in projects, when I was using OTUs, I would have like 70,000 different OTUs. And of course, that seems like a crazy number of different taxa to have in some, you know, 20 different samples possibly. But when you use ASVs, I was going down to like maybe 6,000 different ASVs across these samples. So OTUs are kind of introducing often a lot of weird things because you're looking at just this like one base pair difference possibly between it. Yeah. So this was a comparison a few years ago by Jacob, who was a previous PhD student with Morgan. So he ran all of these different denoisers. So if you look here on the left, we have the kind of expected community. So these were using a mock community. You do get slight differences depending on which of the different denoising pipelines you use, and particularly you get different numbers. So as I was saying, you often get a lot of different OTUs, a lot more than you would expect based on what is in there. And especially as these come from, I think, probably 20 different taxa in the original sample. So you can see how these really will inflate any differences in diversity that you might be looking at. So after you have denoised, you end up with some kind of an OTU or an ASV or a feature table is what we usually refer to this as. And when you hear the term feature table, what we usually mean is that we have a list of kind of sequences or taxa, and we're looking at their abundance across different samples. So you'll hear feature table used kind of regardless of whether you're looking at genera, species, ASV, functions, like all of the above just seem to kind of get grouped into a feature table. And here, yeah, you're looking at the number of reads typically for each of these, in this case, OTUs or ASVs in each of your samples. Of course, this is what QIIME gives us as an output. So each of our ASVs is kind of summarized with this MD5 sum. So anyone familiar with an MD5 sum, you often have it when you're downloading packages. It's a way of checking whether something is identical to something else. And there are programs that kind of generate this for you. And this actually means that each of these corresponds to a specific sequence. And this would be identical between different studies, but it's a little bit difficult if you're talking about the different taxa in your sample to say, oh yeah, I mean, this 708851F whatever is abundant in just these 3 samples, whereas this 7F02 is in my other samples. And so usually what we will do is we'll just assign each of these a name or a number, ASV1, ASV2. I did actually review a paper a while back where they just used this MD5 sum, and it was absolutely horrendous to try and read. They really didn't want to change 20 names, but it was, you're reading a second paragraph and you can't remember if it's the same thing as they were talking about in the previous paragraph. So yeah, it's, while not completely necessary to change something like this to a number like ASV1 to A, B, C, whatever, it makes it a lot easier when you're going to be referring to it in the same way that this is the reason why we give things names, like species names and genus names. So yeah, then on top of these, we also often add taxonomy. So often what we're most interested in is not the number of different sequences that we have, but the number of different genera or species or something that we have. So then we're going to be taking these ASVs or sequences, and we're looking to classify them taxonomically. There are a bunch of different taxonomic classifiers that you can use. There's this kind of ribosomal database project classifier. We have NCBI BLAST, basic local alignment. RTax was another one. And what we're often using in QIIME is the naive Bayes classifier. This kind of a machine learning based classifier. And often what matters more than kind of this part is the database that we're using for comparison and what we're using to assign taxonomy to things. So there have been a lot of different databases, and I have another slide on these in a second. But essentially what we're aiming to get out of this is some kind of ranked taxonomy where you want to know, if you're looking at 16S, is it a bacteria or archaea? What phylum does it come from? What class? So you'll usually at the end of this have something that looks like this. So for each of these ASVs, we have their full kind of taxonomic assignment. So, in the case of this first one, it can be classified confidently down to this genus. And then QIIME will give you a confidence score as well. So this 0.99, that's great. 1 is the highest it can be. That means you're 100% certain that it definitely is that taxon. And you'll find, you might have this species classification, and that's 0.83%, 0.83 confidence. And typically this is kind of, you're going to get lower confidence as you kind of get more resolution. And it's kind of up to you how you feel about how confident you're going to require those to be. Because I think each of these will allow you to say, if it's below this confidence, then take it up to the next taxonomic level. So you might then be saying, okay, it's got to be this. I'm looking at the genus, not. Yeah. I don't know. Honestly, you often kind of have a quick look through these and see if they seem reasonable and then move on. Like I usually for Amplicon sequencing, when you're looking at a short region, we typically aren't looking below the genus level. Because once you're looking at the species level, you'll find like maybe 20 or 30% of your ASVs actually have a species level classification. And so I think that's why the confidence isn't always as important. I think as we move more to like full length 16S sequencing, I think it might become something that we need to look at in a bit more detail. But at the moment with shorter amplicons, it tends to be that once you're looking at the genus level is pretty certain anyway, unless you're looking at a very kind of novel environment. Hopefully that's another kind of hand wavy. There isn't really one single answer, but yeah, higher is better, but what you choose as acceptable is not necessarily what I would think is acceptable. So yeah, it's difficult. But yeah, the reason that we want to do this usually is because it's a lot easier to remember. Taxa become known for their functions, particularly when these are conserved within different groups. And this means that we can group things together at different resolutions. And so typically you'll also find that if you look at the prevalence of a species, so how many different samples that you have that a particular species or ASV occurs in, it's often very low. You might only be finding it in 5% of your samples. And so then if you're looking to find taxa that might be good indicators of something, or, are they more abundant in cases versus controls, you can't really do those statistical tests when it's only found in 5% of your samples. And so often we're looking at the higher taxonomic resolutions just so that we're at least finding them in a lot of different samples, and you can actually do these statistical tests with them. So yeah, many different taxonomic databases. RDP was probably one of the first ones. So this stands for Ribosomal Database Project. This includes bacterial and archaeal 16S and fungal 28S, as well as ITS. They most closely follow the NCBI taxonomy. So that is one that is kind of, there's a massive NCBI database of taxonomy. And it's kind of any new genomes that get submitted, or that's the taxonomy that kind of gets followed for it often. But it's not necessarily revisited very often by taxonomists. So you'll find that people that are very into taxonomy often will be using something that isn't NCBI taxonomy. They brought out a classification tool for it with it. Although I was looking for it the other week, and it looks like the website no longer exists, although you can still use that classifier. But it was last updated in 2023, and it has about 25,000 sequences from kind of close to 20,000 species, or about 4,000 genera. The silver database has 16S and 18S, as well as 23S and 28S, so for bacteria, archaea, and eukarya. It was initially preferred by Mother, and it has been used kind of predominantly by QIIME for quite a long time as well. So it is used very frequently, microbiome analysis. They also integrated their taxonomy with GTDB, and they have taxonomists that work with them that kind of verify each of the classifications in a way that doesn't really seem to happen with the NCBI database. They also release a phylogenetic tree with each version that you're then able to use when you're wanting to kind of insert your sequences into a tree instead of building a de novo tree each time. But that's going to be covered a bit more tomorrow morning. The most recent version was 138.2, and this has kind of over 2 million SSU sequences from over 17,000 bacteria. And then the final database that is very commonly used is the Green Genes database. So this was used quite a lot initially when it came out, but they didn't really update it from 2013 until 2 years ago in 2023. And so by like 2020, 2021, we were like, well, we can't really use this because we've done so much sequencing since 2013. You know, many of the microbial taxa have only been introduced into databases in that time period. So it wasn't really feasible, but they did eventually update it. And I think what they were aiming to do there is where silver often only has a 16S sequence. So you can't really use it if you're looking at like metagenomics as well. You would end up with different taxonomy between the two methods. In Green Genes 2, they came up with they've got genomes as well as the 16S sequences in there. So theoretically, you can use this for both 16S as well as metagenomics, but it only has 16S. It doesn't have something like 18S or ITS, but it does have a lot of sequences. It has about 21 million sequences. There are also many, many other databases. So for some specific applications, there may be different databases that's kind of recommended you use. So there is a human oral microbiome database. In our investigations of saliva sequencing, we found that this usually gives better species level classifications than one of the bigger databases. It's much reduced. It's only about 1,000 taxa in there. But if you've got a relatively well characterized system like the mouth of people, then we've probably characterized most of the taxa that are there. So it's relatively reasonable to use something like a specialized database for it. This is a metagenomic paper that I've got at the bottom here, but it's just kind of a cautionary tale, I guess, where using these specific databases is not always good. So this was kind of an interesting story where one group of researchers from a university published a paper where they, I can't remember what it was that they identified in all these different people's stool samples. It was like, Morgan, do you remember what it was? Okay, but anyway, they identified a lot of kind of surprising taxa in these gut microbiome samples. And then this second group of researchers from the same university published this paper later where they used the same samples and they used a database that only contained sequences from different frogs and reptiles. And they kind of identify all of these frogs and reptiles inside these stool samples from the people. And it was just kind of demonstrating that if the only thing you have in a database is kind of something ridiculous that wouldn't necessarily exist there, you're probably still going to find it with the sequencing methods because it's the only thing you're looking for. And you can find some sequence similarity between a lot of different things. But if you actually had things that match it much better within your database, then you'll find that. And so there is much less of an issue with something like Amplicon data than it is with metagenomics, but it's still worth keeping in mind that if the only thing you're kind of allowing yourself to find in a sample is not likely to be there, you may still be identifying it, but that doesn't mean that it's correct. And so we will, even if you want to use something like a very specific database, I would usually recommend a kind of sanity check after, where you at least search a few of your sequences in something like BLAST. So that's an NCBI tool that it allows you to kind of just search your, like paste in your sequence and search it against their entire database. It's kind of too, it would take you too long to do this on all of your sequences, and it's not necessarily the best taxonomic classification that you can get. But because it's a very comprehensive database that you're comparing it against, it's a really good sanity check that you're at least getting something similar when you search it against that database, as well as what you've been given from your taxonomic assignment results. And so then for different regions, you have different databases. So for 18S, the database that you'll be using if you choose that data set for the lab today, the PR2 database. And so this is one that focuses on protists, but it also contains metazoa, fungi, and plants. It has about 57,000 taxa and over 220,000 sequences. And then if you choose to look at the ITS dataset, then the Unite database is what we're usually using. This is for the ITS ITS region. There's close to 4 million sequences, 239,000 fungal. You'll see SH on it. That stands for species hypothesis. I don't know a whole lot about fungal taxonomy, but this, I guess, is kind of getting at the fact that they don't know as much about it as you do about the bacteria and archaea, and so this is how the taxonomists have chosen to deal with this. But within this, it's not only fungi, it's also other eukaryotes as well. And so this is kind of their species hypothesis graph for fungi. You can see that most of them come into this classification, and similarly for the eukaryotes, most of them come into the Viridae Plantae. So after you've done the taxonomic assignment, you'll usually do some more filtering. So we typically will filter out what we call bleed-through ASVs. This is a bit less of an issue in the newest Illumina sequencing, but previously, and I think still a little bit, some of the sequences that you pick up in each sequencing run are actually from the same indexes from the previous sequencing run. So based on Illumina's reporting of this kind of problem, we tend to filter out about 0.1% of the mean sample read depth, because that's what we think is typically going to be coming from the previous run, possibly. So it's important to kind of take those out, because we don't want to be finding these weird taxa that actually wouldn't be in our samples. It's just whatever was sequenced in the previous run. Then we will usually filter out any contaminant ASVs. So for 16S, we're typically not wanting to look at mitochondria or chloroplast DNA, so we'll filter those out. So anything that our taxonomy has said is mitochondria or chloroplast, we'll filter that out. We also often will filter out things that don't have a classification below like the phylum level, for example. That would depend on your environment. If you're looking at, human sequences, then it's pretty unlikely that there's a brand new phylum that you've just found in your samples that no one had ever found before. At this point, maybe 10 years ago it was different, but at this point it's relatively unlikely. But obviously, if you are looking at a very novel environment, then filtering out novel phyla may be a bad thing to do. But it's often possible that these phyla that are unclassified, it might just be a kind of really crappy DNA sequence that got in there somehow. You don't necessarily want it. And then, yeah, you're often filtering out any kind of eukaryotes or non-target. So as we kind of said before, looking at some of these primers, while yes, bacteria and archaea might be what we're interested in, a lot of, you're still going to be picking up a reasonable amount of these other groups. And depending on your sample type, you may have more or less of the DNA in your original sample that came from some of these other groups. We also often will filter out, for example, samples below a certain sequencing depth. You might decide this based on how much sequencing you've asked for. If you have 100,000 reads in most of your samples and one sample that was processed in the exact same way that only had 1,000, that may just be that the sequencing didn't work very well, something else happened, you had a contaminant, whatever it may be. You're probably going to want to remove that sample from your further analysis because it is clearly quite different from the other ones. And you also may be removing ASVs that are low prevalence, for example, in below 10% of your samples or something like that, or species or genera that are present in not very many of your samples. And that will depend on how similar you're expecting your samples to be. If you had 3 biological replicates of something and you've found something that's only in one of those, then maybe you don't want to be looking at that at all in the future. Depends how much variation you're expecting to see. If you're sequencing a population of people, it may be that you don't want to remove anything like that because you're expecting that people can have more variation between them. Okay, so finally, I just wanted to touch on 16S copy number. Sometimes people will do normalizations for 16S copy number. And sometimes it's recommended that you kind of normalize your data afterwards in this. But 16S copy number varies a lot between different bacteria and archaea. So this was a study from quite a few years ago now, but they found that there was a variation between 1 and 15 copies per genome, with an average of about 4.2. And this does vary somewhat by different phyla, but it's not consistent enough that you can assume if you're looking at proteobacteria that you're going to see always 316S copies and you can normalize in that way. And there's actually more variation with genome size than there is with 16S. No, sorry, other way around. There's more 16S copy number variation than there is in genome size between closely related taxa. And even within different strains of the same species, you can see different numbers of 16S copies. So you can see why it might be difficult to normalize for 16S copy number. You also find that 16S diversity increases with copy number increases. So often the 16S copies that a particular bacteria has will not each be identical to one another. And so sometimes this is why you might be seeing that you have a lot more ASVs than you're expecting if you sequenced a mock community or something like that. Yeah.
That data in databases, so that post-analysis takes this stuff into some, or is it?
Typically not.
No.
I think just because it's so difficult to control for. So yeah, this one, the next one says, there are a few tools that try and do this. And even in PyCrust, which Morgan developed, I've done some work on as well. It does aim to do this, but it's not straightforward enough that it's kind of routinely done. I guess if you had a kind of defined community that you were just trying to modify in some way, maybe it would be useful to do so that you could get back to the abundance. But typically it just doesn't, it's not consistent enough that it's easy to do in practice because you just can't estimate it that well. And so, yeah, this was still a few years ago now, but I don't think this has changed since then. I wouldn't say that it has become a solved problem.
Not an issue with DPS.
I don't know how much that copy number varies. I mean, you have a lot of very big genomes in some of those. I'm actually not sure. I would imagine it's still an issue, but I'm not 100% on that. Yeah.
What's the benefit of writing for copy number?
It depends what's most useful for your community. If you want to, so typically, if I were writing something up about a 16S study that we're done, I would describe something as like the abundance within my 16S community. And that is not the same thing as the abundance within the original community that you started from. So if you were able to normalize it efficiently enough, then you would effectively be able to know how abundant that was in your original community versus within the genes that you sequenced. Does that make sense? Yeah.
I think in the primer, I was talking about combining things with qPCR.
You can. It's like our journal club, we looked at a paper the other week that was trying to get at absolute abundance. I mean, obviously they say it's solved. I disagree. They showed that it worked in stool samples, which are very high bacterial biomass. And they said, if essentially that the amount of DNA in their samples originally was really highly correlated with the actual counts of bacteria within those samples. in my opinion, they only showed that within those stool samples. If you look at something that's not like already 99% bacteria, I think you're going to have a lot more issues when you don't know how many of your reads to begin with were from, like if you're looking at a person, you don't know how many of the reads were human to begin with. What proportion? Did someone take antibiotics? Did it change between 3 of your samples? Like, I don't know. There are methods that people use to try and get closer to that original number, but I think it's complicated by so many different things. And I mean, it assumes that you've extracted all of your DNA efficiently as well. If you're looking at a biofilm versus free living bacteria somewhere, that's going to be really different. You know, maybe if you've got an entirely planktonic sample, you could do flow cytometry to get quite an accurate count. If you're looking at a biofilm, you probably can't even efficiently detach everything from that surface. So I think it depends. In some systems, maybe you can get much closer to that. I don't think as a whole, it's kind of a solved issue. Anyone else? Okay. So that was kind of the end. Any other questions?